Welcome to Brain Ponderings, your window into the world of brain research with conversations with scientists at the forefront of their particular area of this discipline. Today, it's my pleasure to welcome Francesco Papaleo. He's uh,
I
he's a tenured principal investigator at the Italian Institute of Technology in Genova, Italy. Genova.
Genova,
Yeah,
correct.
no V, but it's pronounced like a V.
Yeah, it's a bit.
And is that kind of the equivalent of MIT in the United States?
Yes, the let's say smaller, but the idea was that, or like the Max Planck in Germany.
Ah, okay.
Yeah.
And to introduce you to what Francesco's been working on and what he's going to talk about today, I'm going to read some quotes from recent papers.
Okay.
Okay. And he's going to talk about brain-brain interactions and there's evidence that there's synchronization of neural network activity, maybe in my brain and Francisco's brain right now. And some that relates a lot maybe to our emotional state. And so here's the quote. Interacting brains operate as an integrated system with neural dynamics co-evolving over time. Neural synchronization across brains has been observed in a range of species, including humans, monkeys, bats, and mice. The interneural synchrony has been proposed as a potential mechanism facilitating social interaction by enabling the functional integration of multiple brains. Individual responses such as emotion processing or decision making are adjusted and updated based on information that is continuously exchanged among the interacting partners. So that hopefully that kind of summarizes what you'll
Uh-huh.
talk about.
Yes, yeah. I don't know. You want me because we started. This is what really we are doing now. What we have been doing recently, indeed, is been focusing on how, in general, emotions shape our way to interact with each other, so to respond to others, to understand others. And until most recently, let's say the published work mostly was on checking one single brain at a time. But
Ah,
of course, this is a partial. It's only one small pieces because it's in the common knowledge that when we interact with each other, we continuously adapt our behavior and our way how we respond depending on the feedbacks from the others dynamically throughout time. So in this sense, yes, this is what we mean by thinking about multiple brains interacting together as a single unit. That, of course, they interact and they respond to each other. So this is, of course, is not a novel idea. So, there are a lot of evidence and experiments done in humans. This hyperscanning, no? So checking multiple brains interacting together, and
so
yeah,
what is hyperscanning?
hyperscanning is basically checking and recording the activity of multiple brains simultaneously.
Okay.
So I think this is the ultimate goal. I think at the beginning, we might, some of the initial work was what probably a lot of people knows about these mirror neurons, no? So this that was still at the single brain level. So when, the same neuron will be activated when you see somebody doing an action or when you do an action itself. So this was the famous work started from actually an Italian, Giacomo Rizzolatti here in Parma. So that inspired us as well and a lot of other people. But still, that was a single brain. So this might not catch the complexity, of this dynamic interaction over time. So in order to do that, different.
Francesco, this, so this mirror brain, so the initial experiments so the idea is if you see someone who's suffering and in pain, this is one example, then uh, the networks that are active in that person who's actually in pain, kind of similar networks will
when you
become,
receive that pain yourself.
yeah, yeah,
Yes. So that was again initiated by Rizzolatti, then it's been now pursued by so many other labs. For example, uh, uh, major labs that are working on this, Christian Kaiser, Valera Gazzola, and all these other people working on these events. But then again, it was one single brain. Then a lot of people started to ask, okay, but in order to catch really these interactions, no, we need to record at the same time
yeah,
two real interacting brains. And so here a lot of human researchers started, with the available techniques. So I don't know, double fMRI scanning,
mm,
double EEG, and so on. I think, yes, big names are Uri Assom, Guillaume Dumas, and all these other people. They started this hyperscanning that I was saying, and so many others. So this has been increasing over time. And I think the interesting and I don't know, still debated evidence was that they started to see some sort of synchronous activity, between interacting brains.
yeah,
Uh, the question is still there if it's just a consequence because they are interacting and doing the same thing, or it's something more that modulate their behavior. So in this sense, I think causality will be needed. There are some groups, for example, in our institute, there is Giacomo Novembre, is one of the experts on this, trying to, in humans, to try to impose some neuronal activity during these hyperscanning experiments, try to see if this indeed is causative in modulating their reaction. But of course, for humans, we are limited by the techniques that we have, and also let's say, the deep understanding of this mechanism is more superficial.
yeah,
This is why a few years ago we started to think about if we could leverage uh, all the potential that we have now for in mouse research, where the technology for this mechanism is much more evolved, and they actually keep on evolving every day. And in our case, we were already prepared because we were already have been developing these new tools that open. It's just a very simple paradigm but that was convincing everybody as ourselves that we could address, even in a mouse, the ability to discriminate and properly respond to altered emotions in others. So this, I think, was a very funny and amazing story that we have been working on. It's been um, entertaining us for quite a bit now, uh, because and I think this started from the talking a lot with the clinicians, that every time they were saying, "Oh, all these patients." Or humans, a lot of research also in healthy subjects is based on our ability to recognize and react to emotions in others. And this was completely missing, let's say, partially missing, as approximating this way in the mice. And we started to generate a simple task paradigm that could address this. And now a lot of people, of course, have been implementing it, validating it even further. So I think we're in a moment that we have a new toy, that we can explore further and further. But at the beginning, we were checking only one single brain. So how one single brain was interacting to emotions to others. And now in the last years, said we've been evolving and trying to really record simultaneously multiple brains, even in mice, no?
but the yeah, so in humans, there's a lot of issue. Right, you can do fMRI. The resolution, spatial resolution, isn't that
Yeah.
great, and even the temporal isn't. I guess EEG doesn't really. I mean, you can say, is there, like, I guess, synchronization of the amplitude of a particular frequency of oscillations,
Mm.
like whatever gamma or the lower end delta. Um, but in the mice, in theory, you can, not in theory, in practice, you can implant electrodes,
Yeah.
record from individual neurons. You can even apply optogenetics. Um, one thing I was going to ask you before I forget it. So the very first podcast I did was with Joe Ledoux.
Hmm.
He's very famous for his,
Yes, I know him very well. Yeah.
yeah. And I asked him, of course, we're talking about emotions. He focused mainly on fear and the amygdala,
Yeah.
and was really kind of pioneering in that work. And so there's other emotions, right? Whatever, sadness, disgust, um, joy, happiness, or whatever, anger. And so I asked him, and he worked on animals, mice a lot. So I asked him, well, what about these other emotions in mice? And I said, well, it seems like pets, if you have a dog, for example, particularly dogs, cats somewhat, um, just kind of anecdotally and non-scientifically, I guess, it seems like for example, when I grew up in Minnesota, we always had two dogs. And and then, a couple times one of the dogs died from old age. One actually got hit by a car. But anyway, when that dog died, it was very clear the other dog was very sad and kind of depressed mood
Hmm.
and so on. And
So what
yeah, and
was
then
the
and
answer
then
of
when and
Joe?
then when they're happy doing something, their ears stick up. We also had horses. I grew up on a farm. When they're happy, or at least, uh-oh. Okay, we're okay. We just switched sides. I don't know what happened. It's okay. Um, but anyway, you know, horses, when they're kind of happy and attentive, their ears are forward. When they're angry, their ears are back. And so I asked Joe about, and he said he was very careful. He said we shouldn't ascribe human emotions to animals. But I look at it in the opposite way. We're so we're so similar. It's certainly at the molecular and cellular level, and even circuit level, for the most part, brain circuit. We're very similar to most all mammals essentially. So I look at it the other way. I think we should assume the animals have those emotions until we prove otherwise.
Hmm.
What?
But I mean, my take, my approach to it, let's say, is of course we cannot ask a mouse or a
Yeah,
rodent or a dog how
yeah.
it's feeling, what
Well, they
is,
don't
and
have
we
language, but still,
exact, but we cannot be sure that they can be conscious of a specific emotion or label it. Okay, but in our definition, so emotions is like a reaction to some event, mostly acute event. And in this sense, emotions are very highly conserved because what they express a state that needs to indicate and express outside what somebody internal state have been receiving. And I think I say this is important because it's fundamental for each animal to understand this in others.
yeah,
Otherwise, they will not be able to survive or to, I don't know where is the food or escape for a threat.
yeah,
So,
right.
in this sense, I believe these processes are conserved.
Yeah,
Then, of course, if we instead mean that, okay, me, Francesco, I understand that you, Mark, are happy in this moment, this we cannot be sure that they label in our same way or they perceive in the same way. But they will respond accordingly to the environment
yeah,
and to a huge evolutionary pressure that have been, let's say, co-evolving emotions with the social behavior because we are tuned to this since we born innately. No, the newborn will look at the mother and see will react
Yeah.
differently if the
Yeah.
mother
Yeah.
is happy or not.
Yeah.
So I think these are really, this is why we focus this a lot on it, and we think it's valid throughout evolution because they are very important. Then there are more advanced, even now, labs that have been indeed showing that in different animals, including mice. There was a recent paper now a couple, 2, 3 years ago from Nadine Gogol and Max Planck, was showing that an algorithm can track the different expression of the face of a mouse reacting to, I don't know, the disgusting water or a pleasure, like sugar, or some stressful conditions. And so this could then be considered as an expression of emotions. Then emotions, of course, is not only the face, it's all the brain, it's all the body. But yes, they do this. So,
Uh,
and again, because they need to express this to tell outside what's going on, to give information to the others.
yeah, I was thinking of another example of that. So we have a cat, right? And uh, cats, and our cat likes a lot of attention from humans. He's always likes to be around people. And he'll when he's close to you or you're petting him, he'll purr,
Yeah.
make this sound, right? And it seems pretty obvious he's feeling pleasure
Yes, yes, yes.
when he's purring.
Yeah,
So
yeah, because again, in this sense, we need to think your two brains are
yeah,
connected, because if it was not pouring, you will not probably keep on cutting them.
yeah, yeah.
And so this is what I say. Indeed, the real big picture is to consider all the brains interacting together
Okay.
and see how they do it. In this sense, I want to mention, I think also one of the first evidence that this interbrain connectivity were even visible in mice and bats. There were these two labs, one of Waits Hong in US and the bats from Israel. Really, I think show that indeed even in animals you can have this phenomenon. No.
Yeah.
Then of course, as you said, we still don't know what are the specific neuronal mechanism underlying it, and still if they are causative or just a consequence. This is still something that we are working as well on. But yes, this is another proof that these processes are well conserved through evolution.
You mentioned the mother-child relationship, the bonding, strong
Yeah,
bonding, and the evolutionary importance of that, which is obvious. But so in humans, is there, I guess you'd say, stronger synchronization between, say, a husband and wife, and more closely
related people. Yeah,
socially related than, a stranger, like me and you, we're in a
yeah.
sense strangers, although we're not strangers to neuroscience, but we're personally strangers. So that kind of study, I think, has been done, right, in humans?
Yes, yes, there are a lot of evidence showing that, I don't know, romantic couples or housemates and college,
Yeah,
they show an increased synchronicity of their
yeah,
brain activity. Now, the question out there is still, is because they know each other more, so they
yeah,
are more
yeah,
tuned to each other, or they notice more, let's say, the cues that each other engaged on, or is a process indeed that builds up independent of this physical thing. And so I think this is also a big question. And I think also a big question is for completely stranger people. I don't know. This is something that we want to work on.
but what if
Okay.
you have, so I mentioned you and I, right? So what about two strangers that have a common interest or a common affiliation with a group, uh, I don't know, a political party
Yeah,
or
yeah.
religion, if you want to get kind of really strong group relationships. Have anybody studies been done looking at interbrain synchronization? And because there they don't have
No,
any
as far as
personal.
I know, not systemically. So but this is also one of my, I think, questions for the future. No, for again, for completely strangers or belong to two same groups, or I don't know, you have some with some people you get along better immediately. No, this
Right.
is there something into your brain that will influence this
Yeah.
between your brains?
Yeah.
Uh, I mean, I mean, a friend of mine, John Cryan, sometimes he say, no, that is also the gut feeling, no, even more.
Yeah.
So, like the microbiome can all even have a role on that. But yeah, I think that the brain, what in the brain is what I would like to study this.
Are
If
you
there is
still
some,
are you still doing okay, wait. Well, let's get into the mouse research and exactly what you've been doing. But first, are you still doing human work in
so
parallel?
yeah, we were in our lab since actually since I started at the NIH, when I was at the NIH in US,
Yeah.
my work has been trying to combine as much as possible mouse work and human
Yeah.
work.
Yeah.
Of course, with each limitations of them, but with synergy between them. And so, yes, when we can, we always try to have to
Yeah.
build these
Okay.
bridges between mouse research and human research. I mean, the last publication that we have been doing, we have been trying to combine them directly.
Oh,
We are keep on doing this. Of course, sometimes we do. Most of the times, also we do in collaboration with experts on this field. For example, now we are working a lot for the human parts of the inner brain with Giacomo Novembre or with others. Depends on but we always try to combine. This is also why we went in mice and we saw, okay, there is a gap in the behavioral assessment, no, between mice and humans for these emotion recognition abilities, no?
yeah.
Because a lot of studies were done in humans. Instead, in mice, most of the time, we were relying on very basic social interactions, you know? So this is why also we try to, I mean, knowing and talking with the clinicians and human researchers, then
Yeah.
we
Yeah.
can also figure out what are the gaps and trying to fill these little gaps, you know, with a brick at a time, no? So we cannot solve everything immediately, but we try to advance a little bit on this.
Okay. So in the mice, what is the experimental paradigms you're using and what kind of recording methods? Can you get into the technical
Yeah.
approaches?
So the behavioral paradigm is very simple. The test setting is very simple. So you put uh, one mouse that we call observer in front of two other mice that we call demonstrators, which they don't see each other, these demonstrators. They are in two corners of a small cage, and they are in their own, let's say, room, small rooms where they can exchange visual, olfactory, auditory, touching cues between each other. And then you see this observer how will explore and interact with these two demonstrators. And usually we manipulate in a mild way the affective state, so one of the two demonstrators, in order to induce a positive or a negative affective state. And then we see the discriminatory behavior and the reaction of the observer towards these two demonstrators. So I think the key point is to have the choice between them, because again, we cannot talk with the mice, so we cannot ask them, are you seeing a stressed mouse or an happy mouse or whatever. Instead, in this way, we can infer how they react and what they choose to interact with. So this we validate in different ways. We know now that this sort of discriminatory behavior depends on olfactory and visual cues in mice. Instead is less dependent, at least in adult mice, from not dependent actually from auditory cues. We even have some new data, not yet published, that tactile stimuli are not so important. So, for example, whiskers, contact, and these things are not so important. Instead, the visual and olfactory, yes. Why I say this? Because we usually see that if you induce a mild altered states, so even positive or negative, usually the observer mouse spend more time sniffing that emotionally altered mouse compared to the neutral one. And this happen in very few seconds, up to 2 minutes usually. If we do, for example, this task in complete darkness, they don't see each other, so no visual cues. They still do it. So they do it. So indicating that vision is important. But we did try, we published the showing if we show them instead the 2 videos only of these two demonstrators, so the videos of mice.
Yeah.
Of course, they are less interested in videos compared to real mice, but they still discriminate between the two. So visions is important. And another proof I think is interesting that if we give them only odors, so for example, we instead of putting them in front of mouse, we put the cotton swab that we swab on the two demonstrators with the same odor, so neutral odor and emotionally altered odor. They will discriminate them, but in different way as if there are the mice. For example, a mild stressed mouse, the observer mouse will go more towards the stressed mouse. Instead, they will avoid completely the stress odor if there is only the odor.
Interesting.
So I think this is very interesting because it means there is a ethologically valid way to respond to emotion. So if there is only the odor, at least this is what we interpret, this is a signal of alarm. So I need to escape that. There is something dangerous. If there is a mouse instead that is mildly stressed, maybe I want to go there to help or interact or buffer the emotional state. Instead, if we give the odor, for example, of a pleasure, I don't know, like giving a reward or something, they will go even more to the others, even if it's just odor, no? These are just examples as this simple test can be validated in different ways, can give you different information. We can extrapolate, let's say, our interpretation of these behaviors.
Okay, and then you're recording from their brains during this interaction?
So yeah, the idea was to leverage on all these techniques that we have available in mice, are much more advanced in mice. Of course, there are many other species in which these techniques are being developed or adapted, but in mice we have much more. Still, they are being developed continuously. So what we record in different ways. So we have been doing with electrophysiology, so single unit electrophysiology with electrodes. We have been doing, and so this give you the, let's say, an advantage of having being fast, fluctuating signal, or whether we cannot be sure exactly of what kind of neuron we're recording. So,
so maybe at this point, what can you summarize in the human data, for example, with fMRI, what neuronal circuits are most synchronized, and does it depend on the
depends
emotional?
on the context.
Yeah, okay.
What I think a common, this is why also we are more interested in that, let's say, a common most, we reported this evidence also in our recent review. There are some good reviews from Leonard Schillerbach on these specific circuits that can be seen to be more synchronized between humans. We see a consistent findings mostly on the level of the prefrontal cortex, seems to be one of the major hub, especially the anterior cingulate cortex, to be involved in this multi-brain synchronization. This is why also we start from that. But I would not exclude, I'd say the idea is not a single brain region involved. Of course, for social interaction, the entire brain is involved. So it's true with, for example, with EEG, mostly you see superficial, no? Regions, so you get more information from that. So this, I think, will be needed to be enriched more and more
So a lot of
on
focus
this.
on prefrontal cortex because of its roles in goal-directed behavior, decision making, impulse control, that
Yes.
kind of
And for this initial,
top down process.
yeah, and for this initial evidence that indeed is for multi-brain synchronization seems to be
Okay.
a repetitive hub that is been reported more
Okay, so
consistently
in
in
the mice,
humans.
you're putting the electrodes in the prefrontal cortex.
Yes, or as I was saying, um, to have a better idea of what kind of specific neuronal subpopulations
Oh,
are involved, we use a lot microendoscopes, so using calcium imaging, so
oh,
calcium
cool.
fluctuations. So we are using this a lot on microendoscopes. This give you cell resolution activity with also the ability to recognize, okay, this is a specific subgroup of inhibitory neurons or pyramidal neurons, or etc. In this sense, we have been showing still at the single brain level that, for example, for emotion recognition, a subpopulation of inhibitory neurons, so-called somatostatin positive cells,
Yeah.
are very much important in this ability to uh, recognize and respond to altered states, emotional states in others.
Oh, interesting. Well,
Yeah,
yeah, and that
this
maybe that makes sense in terms of, even going back to more crude things like the EEG, right? So there's quite a bit of evidence these GABAergic inhibitory neurons play an important role in brain oscillations, right?
yeah. It's
Um,
kind of like they are orchestrating, but here even we have been seeing that different GABA neurons within the prefrontal cortex
yeah,
might have different
yeah, yeah,
impact in these social behaviors. For example, another class of neurons, inhibitory neurons, these parvabum in positive cells,
yeah, yeah,
were important instead in social interaction when no emotions were involved, no? So in basic social interactions. Or more recently, we show another subgroup of inhibitory neurons, these inhibitory neurons expressing corticotropin releasing factors,
yeah.
CRF.
I saw that
The,
paper. Yeah, that was interesting.
uh, these were instead kind of, we think, storing a memory, an emotional memory in the observer that then were important when they were interacting with another emotional, this showing another in the same emotional states. So this all the idea again on the communication between brains, because we all know that we react to emotions in others also based on our own experience.
Yeah.
This was the beginning of our study,
Let me
and
ask you this
yeah,
in case people, because we have like a broad array of people who listen or view
yeah,
these. So CRF is corticotropin releasing factor, and
yeah,
it was initially discovered because it causes the release of
yeah.
adrenocorticotropin, ACTH, from the pituitary gland, which then goes into the blood and stimulates the production of cortisol from the adrenal glands, the stress hormone. So this is part of your stress, uh,
HPA axis,
yeah,
so-called, yes, and the
hypothalamic
stress.
stress axis. So, negative emotions, there's a lot of stress associated with, a lot of times, if you're in a fearful situation or even you're injured or in pain, um, if you're angry, you get activation of this. So do you think that this CRF then, and you're looking at CRF not at the hypothalamus,
and
but
the prefrontal cortex.
prefrontal cortex. So do you think evolutionarily this peptide CRF is having like a fundamental role in integrating the uh, stress responses from a cognitive level, at a
Yeah,
cognitive level?
yeah, this is a good question. I mean, and this CRF is kind of one of my first scientific love because my actually PhD thesis was on CRF, but at that time mostly on the stress reaction starting from the PVN.
My master's thesis was on the regulation of cortisol production in response to ACTH. What was the signal transduction mechanism?
Exactly. And I mean, you know all these peptides, neuropeptides, they works as hormones, but also they works as
Yeah,
neurotransmitters. So they go into the brain. In the specific case of CRF, less than the hypothalamus, but they are also expressed. There are CRF neurons expressing CRF at the level of the prefrontal cortex. There are more in the central amygdala. So again, as a reactivity to the stress. So in general, the idea is that yes, the CRF system is kind of system made evolutionary able to cope
yeah,
with the stressful situations at all the different levels, including the cognitive responses to stressful situation.
yeah,
So it's basically bridging these hormonal reactions with what we decide to do with that.
yeah, yeah.
And
Very interesting.
in this sense, I think was interesting because the question was, I mean, talking with a lot of clinicians and psychologists, there is this evidence that for the same emotional experience, some group of subjects, then they will develop more, they will be more prone to altered emotions in others, what we call sometimes they are more empathic, they are more
Yeah, yeah,
sensitive to.
sure.
But the same instead experience in somebody else could even give the opposite reaction. So when you see that emotion, especially for negative, you will feel self-distress and so avoid them. So what are the mechanisms on supporting this? So this is what our drive for this last publication. Uh, and we think CRF, at least in the prefrontal cortex, is implicated in this process, can mitigate or can drive these reactions. And indeed, as I said, in the same paradigm that I told you before, usually, for example, a mouse will interact more with a stress mouse. If we give the same exact manipulation to the observer mouse before, 1 week, 1 day before, again, this is not trivial. It's the same exact manipulation. Only if they see the same exact in the other, then they might shift completely from approach to avoidance. I think that this is very cool because, and it was not trivial because they, despite not seeing the trigger of this emotion, so what the event that induced that emotion, they can understand much more than we think because they respond only if they see the same thing that they went through before.
Yeah,
And this was very cool, consistent, and they might completely shift, or some they can instead go even more. So I think this was cool. Yeah,
this was your Nature Neuroscience paper?
yeah. This was our last Nature Neuroscience paper.
Yeah, yeah. Can you t talk about that? All right. So this is the one where Federica Maltese, the first author.
Federica Maltese is the first author. Yes,
Yeah,
she was the very, very talented students working on it for longest.
yeah, yeah. Can you go in a little more detail on that? You kind of explain the result,
Yeah,
but this kind of illustrate how much work she had to put in, and the other people. But
yeah, yeah. Of course, it
you
was a
know, because
collaborative one.
sometimes people don't realize, how much time and effort goes into all this stuff.
Yes, yes. This was very long, and a lot of effort from all my lab.
Yeah.
But yes, the principal idea was this one. Not, as I told you, we were really interested on understanding what the previous emotional experience of the observer mouse will then influence the reaction to altered emotions in others. This was something that we never did before. Usually we were leaving the observer in normal, undisturbed way. In this case, we started to wonder on this. So the first part, I think the bigger part is this behavioral measurements and testing controls to convince ourselves that indeed they were changing their discriminatory behavior based on their emotional experience, and that emotion was triggered, so they recognize the same emotional states in the other. So
And was
this
this
was, I think,
all, this one question, you probably get in it. This, did you do this one sex, males or females, or have you done both? And are there,
yeah, we do both.
yeah, yeah.
Uh, and until then, we were not finding huge differences
Hmm.
between males and females, but in this case, yes, we found it. We found strong sex-dependent differences, which were dependent on the hormonal status of the subjects. So in particular, we saw that, for example, the dominant males that are supposed to have increased testosterone, they were the one switching more from approach to avoidance based on their own emotional experience. And this, I think, will go in agreement with the uh, previous literature, now showing that dominant mice are more sensitive to stress itself. And so then this will be reflected on being more distressed when they see the stress in others as well, despite being a mild stress. In females, instead, we saw that the estrus cycle during that encounter was indeed protective instead of, in comparison of this self-experience.
Wait a minute, the phase of the estrus cycle?
Yes, when they were in estrus, we saw that they were not having anymore this switch
Ah,
between approach and
ah,
avoidance based on their past emotional experience.
huh, interesting.
Yeah, yeah.
Very interesting.
So I think this is very, add the complexity, but makes sense on when we see all these variabilities also in humans, depending on where we are, how we are. We don't, of course, react all at the same way, but why? And so, here, in mice, you can control better which variable are influencing it, of course.
And in that study, did you do electrophysiology or calcium imaging?
In that study, we did calcium imaging,
Yeah.
we did with the microendoscopes, and so this is the second part of the study. Once we validated all these behavioral readouts, then we wondered, okay, what are the mechanisms? So at the first place, we started with somatostatin neurons because of our previous work, but we were seeing that somatostatin neurons in the prefrontal cortex were equally involved in self-experience, emotion discrimination, or emotion discrimination without self-experience. Instead, we saw that these CRF neurons in the prefrontal cortex are more specifically involved in this memory of self-experience when reacting to others. And I give you, I think, the most striking data. We were imaging them. Meanwhile, not the mice interact with a neutral or a stress mouse, if they have or not this self-experience. So without self-experience, these neurons were more active when the mouse was interacting with a neutral one, and maybe even inhibited when they were interacting with the stress mouse.
Hmm.
So this pattern of activity completely switched when they had the self-experience. So in that case, there were a much higher activation of CRF neurons in the prefrontal cortex when they were interacting with the stress mouse, but not with the neutral. Completely switched. And this was driving then, okay, then here we ask, okay, is this important in driving their choices? and with a different optogenetic manipulation, silencing them, inhibiting them, we were able to show that indeed only when there is emotional self-experience, when you have a relative increase of activity of these CRF neurons, they avoid the mouse that is paired with this relative increased activity.
Interesting.
So this is what we feel. This is what brings us to think that these neurons sort of store this memory of emotional experience that come online when they see the same in the others, and then make them change the reaction.
Yeah, so oh, a technical thing. Right. So are you using genetic tools to selectively introduce the calcium sensitive fluorescent probe
Yeah.
into the CRF neurons?
Yes.
Okay, so you use
So
a,
you have, in this case, you have a CRF Cre line,
yeah,
and you inject a virus that will infect only the CRF neurons in
yeah.
that brain region, in this case, prefrontal cortex. So then we will see the fluctuation of calcium only from CRF positive cells.
In the night, we never used it. So in the 1980s, my lab, I was a postdoc and a guy named Stan Cater's lab who studied growth cone. And we were showing, and I showed specifically, I showed that glutamate controls the outgrowth of dendrites in developing hippocampal neurons and can control synapse formation. And I did some of the first calcium imaging. My mentor was a good friend of Roger Chen, who uh, who developed FIR2, uh, Flow 3, these calcium imaging probes, which were the first things used before they got the genetic
Yeah.
tools. And right, so it, it was really cool. I mean, this is the first calcium imaging in neurons, right? And so we'd see these in these neural networks in culture, you get these spontaneous oscillations, right? And you can see kind of spread through the network and so on. Yeah, so now you're doing really cool stuff where you can look at calcium in vivo in living mice
Yeah.
and look at populations of neurons or subpopulations of neurons and behaving animals. So you're using fiber optics then. You mentioned this
So, or there are two ways. So these are one photon, or now there are even two photon, even three photon microendoscopes
yeah.
that they can check this fluctuation. So in this case, you really see neuron by neuron. Or you also use fiber photometer instead that give you the overall signal. So in that case, it's like the average signal of the entire field of view,
Yeah.
always for calcium fluctuations. I don't know if you want, I can show you some video. I
That'd
don't
be
know.
cool. Let's go ahead.
Let's see if I can do quickly, if I found it.
So you should be able to share your screen if you at the bottom on your
Just to give you a better idea, because yeah.
and for those of you who are listening on Spotify, for example, Francesco's going to show some videos on my YouTube podcast channel, and I expect these will be pretty cool.
Let's see. Okay.
Can you?
Yes, now I will share it. To share, you need to allow me.
Okay. Dang it. Just a second.
So, for example, this was our first work that we did, was led by Diego Scheja, Francesca Manago. So you see here on the right, these are the microendoscope that we use. So this is how big they are. In this case, on the left, you see the single neurons
Wow.
fluctuating. Meanwhile, the mouse is interacting with a neutral or emotionally altered mouse. Um, and this is studied with electrophysiology, but I show you instead this what we are doing when we have all three mice interacting together. And also here you see the simultaneous recording of all three mice, and you see the
Oh, wow.
single neurons. In this case, in this example, are somatostatin positive cells in the
Wow.
prefrontal cortex. In this specific case, anterior cingulate cortex in mice that are interacting continuously, live, freely moving. And then we correlate the activity of all those three mice simultaneously. So this is just to give an example. When I talk about microendoscopes, what you can see
Yeah.
at the level of the single neurons. These are one photon microendoscopes, so mostly you see the soma, the cells, not the dendrites, even though you can a little bit see it.
And then somehow you have to, so you got behavior data, calcium imaging data, and then in real time, and somehow you have to present the data and show the correlation in time, right?
Yes, exactly.
Yeah, yeah,
And that we do in different ways. I stopped the screen sharing. In Yes, there are a lot of different analysis that you can do, starting from trying to see how the two signals, for example, of two brains, they align together, or for example, with cross-correlation analysis, they shift the signal of the two mice in order to see where is the best match, where they will show the best fit. Um, of course, custom imaging is a little bit slower than electrophysiology, so and they are not fluctuating, no? So they have spike and bend down flat, they don't go up and down. So this might create some artifact that you need to control it for. Um, so these are, let's say, the basic analysis. Then, of course, with other more advanced analysis, so this usually we look for support of mathematicians or computational people that can even
yeah,
further analyze, because here you start to have the triple of the data
yeah,
for single neurons multiplied by, I don't know, each mouse at least 50 neurons. So there is a huge amount of data.
yeah.
Uh,
Now, is the next step then to use, for example, optogenetics and
yeah,
like silence the GABAergic neurons or silence the CRF neurons
yeah,
uh, while they're doing this, and then see if the behavior, if they
yes.
don't have the behavior response or that kind of thing?
Yes. This we have been developing and using it for the last years. Uh, Again, all the work on optogenetics that we did publish already so far was in one single brain still. We will hopefully come out soon with the multiple brains.
Yeah.
We are already doing it. But yes, then the idea is this, again, another advantage of using mice. You have all these tools that you can really tightly regulate the activity of these specific neurons, depend on, I don't know, when they are interacting only with the emotional altered mouse or not, for very few milliseconds and so on. So yes, this we have been doing it. We are doing more and more, and that is, I think it's one of the ultimate goal of this interbrain um works, to try to understand if indeed really they are just a consequence of interactions, or if they might really modulate the way we how we interact. But of course, you need to have a lot of different controls. You need to be careful that really to be sure of what you to interpret better the data. So this is what we're spending a lot of time in doing a lot of controls in different conditions. And again, in our case, this simple setup of three brains interacting, the observer demonstrators, is kind of good, because you can really implement a lot of different controls to avoid any artifact or any uh, misleading finding, let's say.
Yeah. Okay, then jumping from that back to humans, and have there been any studies trying to modulate the act, have two humans interacting, uh, maybe using a paradigm that's relevant to this mouse paradigm, and where, because this has a lot of implication that it's, I think everybody experiences this as they get older, in particular in your life, you see people that have a certain difficulty or maybe a disability or, a disease, even cancer or something. And when you're younger, growing up, and you haven't experienced any of these things yourself, you're not necessarily so empathetic because you don't really understand what the person is, going through in their mind. And your work in the mice is kind of addressing that at a very refined level. But then if you jump back to humans, are there any studies where I don't know, using transcranial magnetic stimulation or other ways of stimulating, I don't know, circuits in prefrontal cortex with some of these, you know, interacting individuals, in emotional context? Can one, for example, I guess in an ideal world, everyone would be empathetic to someone who's suffering, right? And yeah, so in theory, is it possible to enhance someone's empathy by stimulating certain circuits or something like that?
Yes, so this is would be the ultimate goal. So for emotions, in the context of emotional context, there is still very limited work in humans. This is also in the last review that we did was actually to realize, explore and realize this. So this needs to be better explored. But for let's say social context without emotions, let's say without strong emotions, there is some work, as you said, using TMS, trying to address if there is causality or not. Again, this, for example, there is some work put forward by Giacomo November, if you want to check better, in the sense, in the context, I don't know, of music
Yeah.
or dancing or on these things, try or tapping with the fingers, that to see if indeed you can increase coordination between the partners or not. So there is some initial attempt towards that. Of course, then with mice optogenetics, yeah, it's more clear, no? Because you can really, because also with TMS, you don't have the neuronal specificity that you probably want to have.
Okay,
Because
so it may be, even though I like rock and roll music from the 60s and 70s, and I don't like country western, it may be possible for me to like country western by brain stimulation when I'm listening.
who knows, probably. I don't know.
Okay, so yeah,
But already I saw, I mean, I really saw some different collaborators that they are working with humans with these neurosurgeons. No, for example, especially for epilepsy, when they are looking for the focus
uh-huh.
of epileptic seizures, and then they have to put electrodes in order to record where exactly it is, in order to better block it or
Yeah.
make a surgery on it. And then they have been doing some pilot experiments on stimulating it. I think that is very impressive, as you can induce also emotions,
Ah,
stimulating specific brain regions. So
yeah.
I saw impressive videos of, you can induce, for example, laugh, just stimulating a brain region, despite you are telling them very sad stories. So yes, this is what when I say that emotions is regulated by so many different parts of the body, including the brain.
Huh. Yeah, I had a podcast on God, I'm sorry, I'm bad with names. I can't remember. But it was talking about Schadenfreude.
Hmm.
That's where someone takes pleasure in someone else's suffering.
Okay, that's another thing.
Yeah. No, but that's a real thing.
Yeah, yeah, yeah.
And
It's a real
yeah,
thing, but yeah,
yeah, and it's not a good thing, I don't think.
yeah, yeah,
But I guess there's some evolutionary reason for that having
yeah.
to
No, the
do
idea also is
with
that that in these psychiatric conditions, indeed there is probably a miswiring of different functioning of specific circuits that then let you react differently to
yeah,
others.
yeah.
So this is our aim is to first understand how these circuits in this social emotional context works,
Oh, yeah, yeah.
and then trying to understand how they work or they work differently in aberrant situations.
Yeah, and there are mouse models that are at least relevant to autism, for example, or
Yes,
the
yes,
Fragile X mice and or maybe schizophrenia, not so much. I mean, you were with Danny Weinberger, or he was the head of the
yes.
NIMH there. You have, I didn't mention it, but you have a adjunct appointment at the Lieber Institute, I think, right? Which was,
Yeah, also because for a period, because I did my postdoc with Danny Wemberger and Jackie Crowley, basically. I was in the middle trying to
oh,
combine
nice.
that.
Oh, that's great
That
behavior.
was
Yeah,
to combine mouse
yeah,
behavior with schizophrenia, human research. I think that was a very nice environment because in the lab there were, a lot of human researchers and also a lot of preclinical researchers. And so then, yes, I was at that time it was kind of crazy. Okay, how you can study schizophrenia in mouse or even autism. Uh, but yes, I think some of this component, like social dysfunctions,
yeah, yeah,
you can address, or even cognitive dysfunctions, you can address, even in a mouse because they are evolutionarily conserved. Then, of course, other things you cannot do it. There is not a autistic mouse or a schizophrenia mouse, no. But these components, I think you can study, and actually, we believe, no, that this might be even diagnostic, no. So because these functions are present in different and common in different neurodevelopmental and psychiatric disorders.
yeah,
So if we understand the basic parts of it, then we can probably leverage on it.
yeah, 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 absolutely. I think it's really cool. And so, yeah, I had Danny did a podcast with me.
Ah, okay,
Yeah,
okay.
yeah. And so the Lieber Institute it's fairly new it's in Baltimore at
Yeah.
Johns Hopkins where I have an appointment. It's actually a lot of interactions with Hopkins and it's a
Yes, yes.
major interactions with them. But it's focused on brain development,
Yeah.
developmental brain disorders, which is a big problem. So what about now that made me think about brain injury. I had a good friend of my son when we moved to Baltimore in the year 2000 when I got the lab chief position at NIA. My son had a good friend who was who moved to DC at the same time, and he was like middle school age, and his friend got hit by a car. He was walking across the street. He had severe traumatic brain injury, and they took him to the Kennedy Krieger Institute. And we went up every weekend, and he was in a coma. Then he came out of the coma, but he couldn't talk. He couldn't move particularly his lower limbs much. But then over a period of months, and even in more than a year, he slowly gets so he could talk with slurred speech. He got so he could walk again, and he even ended up going to college and graduating. So, um, yeah, the developing brain is pretty amazing in its ability to recover from a lot of uh, conditions. Okay, but anyway, so right,
Yeah, no, this
so
for sure. What we and especially, in the more plastic periods, no, from birth up to adolescence, I think is the where it's the most amazing period, when
yeah,
you can really influence more the adaptability, flexibility, and or even where you can have the most injuries that last longer, I would
and
say.
also the prefrontal cortex is
Developing.
it's the last part to
Yeah,
fully develop, and
definitely, definitely.
yeah. Okay, so do you have plans to oh, no, no, now I know why I wanted to mention the brain injury. What about lesions? Are there have do are there people who've had some brain injury from trauma or stroke that affect these mirror neurons or the synchrony between two individuals? Is there any information on that?
I don't know, to be honest. I'm not sure if there have been usage of lesioned patients or traumatic patients in the context of interbrain dynamics. I don't know.
When you were at
Maybe there are,
NI,
but
when you were at NIH, did you come across Jordan Grafman and Dimitrios Kapagiannis?
no,
No.
I don't, no.
Okay. They were doing these interesting studies looking at religiosity
Oh,
and
okay.
neural network activity, and then found some interesting kind of signatures of neural network activity in people who are highly religious versus non-religious. And then more recently, Grafman he has all these access and a lot of data from Vietnam vets who had brain injury. So he has these nice lesion studies where lesions in particular neural circuits can have dramatic effects on someone's religiosity. But I would imagine there's got to be lesions that affect this and maybe some fairly selectively without affecting so much other domains.
Yeah, yeah, probably there are. I'm not. I don't know. I'm not aware
Okay.
of it in a systemic way. But yes, this is all the idea, no? Because maybe we're anecdotal events or
Yeah,
findings that we hope that we will make more sense out of them based on this circuits investigation that we are
yeah,
performing,
yeah,
even if it's only a mouse.
yeah, yeah.
But this can inform better, no, on what's going on in humans.
Yeah. Okay. Well, it covered a lot. I hope you
Definitely.
enjoyed the conversation. I'm going to put in the description section uh, of the podcast, I'll put links to your neuroscience bio behavioral reviews article, which came out this year,
Sure.
I think,
Yeah, yeah,
recently, which is very nice. And then a couple other articles, your CRF1, and maybe a few other original research articles, and your lab page. So if people who are listening or so on want to look in more detail, they can see.
definitely. Yes, yes. No, it's been a pleasure to talk with you, and thanks for contacting me and to have this nice conversation about this topic that we really care about.
Yeah, it was nice to be in sync with you for a
Exactly.
little over an hour. All right.
Thanks so much.
Okay, Francisco.
Thank you.
Yeah,
Bye
bye.
bye. Bye bye. Bye bye.